Try to understand these hormones, right? The first hormone, right, is the gastrin family, right? Before that, let me talk about something called as motilin, right? Now, this motilin is actually responsible for the movement of, I'll talk about it right away because it's a big, interesting topic. So, motilin is a hormone, right? And motilin is responsible for motility. Motility means movement, and the movement in GIT during fasting. Okay, so this motilin is actually produced by something called as MO cells. Okay, it is not present by M cells. M cells are immuno cells. Okay, they have something to do with immune response. Motilin has nothing to do with immune response or immune anything. Okay, that's very important thing to remember. So M cells are microfold cells in Peyer's patches in terminal ileum. Now these are immune cells. They sample antigen to macrophages and lymphocytes. So immune cell M cells, they are immune cells. That's very important. MO cells, on the other hand, are different. They produce motilin. Okay, so this is a very important concept right here. So we have different types of cells: G cells, I cells, S cells, K cells, and MO cells. G stands for gastrin, of course. I stands for intestine. And why we named it I cells? Because it releases a hormone called cholecystokinin. Now, it is very basic in nature that if we eat something, we need the secretion of bicarb and these fatty amylases and fatty lipases and these protein peptidases, these things, right? The things that we use to digest protein, fat, and carbs, right? And it's very normal that presence of these amino acids, fatty chains, will release, right? So basically, cholecystokinin is this middleman, is this middle mediator that mediates the presence of food, presence of fatty acids, and the secretion of bile and pancreatic enzymes. The middleman is CCK. An important thing here to remember is that CCK can act on CCK-A receptors and CCK-B receptors. This is a very important thing to remember. So G cells, they make gastrin, and gastrin is actively responsible for HCl production, and HCl leads to a lot of things. The number one thing HCl leads is converting pepsinogen into pepsin, and that is done under HCl. Why it's done under HCl? Because HCl has this pH, right? And one of the condition in which pepsinogen can convert into pepsin is that you must have a pH which is less than 5. So this is the reason that we don't have active pepsin. That is a very big protein destroyer, or maybe protease, right? That's why this is the reason why we don't have this pepsin in our serum, because our serum pH is around 7.3 to 7.5 or 7.4, whatever you want to think. So that's why we don't have active pepsin just lying around here and there. So this is one of the big reasons. Okay, so gastrin leads to HCl, cholecystokinin leads to fat regulation, digestion. S cells lead to secretin. So S cells are present on the brush borders of intestine, and they produce secretin, and secretin leads to production of HCO3, right? Which is a bicarb, a very bicarb. Now, whenever it's a general theme in the body, whenever there is production of either acid or there is production of any bicarb, there will be a production of its counterpart. For example, if I'm producing some bicarb, I will be producing some acid here and there, so that I am neutralizing it, right? So if my lumen has acid, my basolateral side, the blood side, will have some sort of base, and the vice versa is true. Okay, K cells, weird name. I thought I would find the meaning of this K cell, why they named it K, but it's turned out that it's an arbitrary naming thing, right? They just named it because they don't want anything else to get confused with this thing, right? They didn't want to name it maybe A cells. So they named it K cells, which is very weird. So it makes GIP. What is GIP? GIP is gastric inhibitory peptide. Now, when I'm talking about inhibitory, I'm talking about inhibition. And what kind of inhibition? Inhibition of maybe movement, inhibition of maybe hormone. So it's an inhibitory peptide, and it's a peptide. Okay, it's a protein molecule. This is important. Okay, what about MO cells? MO cells make something called that motilin, which is important for motion. What is motion? Motion is just motion of cells. From where? From, I will talk about motilin later on because it's a completely different thing. Okay, we understand A, nothing, I. Okay, let's talk about gastrin now. Time to discuss gastrin. So, gastrin has two hormones, CCK and gastrin itself. Both of these hormones, gastrin and CCK, are structurally wow, I'm just switching between languages. So, both of the structures, both of the gastrin and CCK are similar in structure. So, it means they can practically bind to same receptors, which are CCKA and CCKB. CCKB are usually on parietal cells, and CCKA are usually in the different places. For example, duodenum, or maybe on pancreas even. So let's start with secretin. So secretion of gastrin, it works via a feedback loop. How? What happens is there is a antral distension. Antral part. What is this antral part? This is my stomach. This is my stomach right here, right? This part is my duodenum, right? This is my duodenum. And what happened here? So what happened here? Okay, my stomach is gone. I'll make another one. This is my stomach, right? This is my stomach, right? Just imagine with me. This is the LES, that is lower esophageal sphincter. For example, this is my duodenal sphincter right here. This is my pyloric part. This is my pylorus of the stomach. This part is the antrum, okay? And this is my body, right? This is my fundus, and this is my cardia. So I hope you guys can remember it from now. So whenever we eat, we have antral distension. The antral part of the stomach expands, and then we have these stretch receptors. Antrum is the lower part of the stomach. It stretches. Now, okay, amazing thing. Now what happened here is that these stretch receptors, they get activated. Once the stretch receptors get activated, we have stimulation of G cells, because G cells are right there, and G cells, upon stimulation, the gastrin is released. Once gastrin is released, it goes and it increases HCl via parietal cells or via histamine. It can happen in both ways. Now HCl changes pepsinogen into pepsin, because changing of pepsinogen into pepsin needs a pH of 5. That is very important. Once we have enough pepsin, it goes and it positively promotes more G cell stimulation, and then we have more HCl, and then we have more pepsin. So this is a positive feedback loop, okay, right here. So in the end, we have protein digestion, and what I have written here, that small amino acid can directly stimulate G cells via chemoreceptors, right? So these pepsins, right, they stimulate the G cells via chemoreceptors. I just wanted to know how it happens, that's it. So this is a positive feedback loop which God has given you for the digestion of protein, right? That's how it works. What about antral distension? What happens when antral distension happens? So antrum, it's the lower part of the stomach, and it stretches. Now we have, what happened here is that we have mechanoreceptors, and they are afferent. We have two types of fibers, afferent, and then we have the body, and then we have the efferent, right? Body of what? Body of a neuron, right here. So afferent is providing information to it. Efferent is this providing information from it. That is efferent, E and A. Okay, so we have these afferent fibers, right? It stretches these mechanical receptors. Then afferent fibers, right? The shape of the mechanical receptor changes, and then they send these short reflexes via enteric nervous system. Okay, and then once these, these short reflexes, these chemical messengers, they go via enteric nervous system, they go and stimulate G cells, and then G cells stimulate gastrin, and then gastrin releases, the gastrin goes to ECL. ECL is enterochromaffin like cells. Now these enterochromaffin like cells, they are the production house of histamine, and usually histamine can work on a lot of things, right? H2 receptors, and they can produce HCl, right? So this gastrin and ECL, they work, histamine and HCl. Okay, what about other things? Well, there is a nerve called cranial nerve number 10, that is the vagal nerve. Well, once we have enough stretch, this cranial nerve number 10, the vagus nerve, will get activated, and then it will release something called as acetylcholine, right? Or it can release CRR, right? Now, what the hell is CRR? CRR is cephalic reflex response. Okay, sometimes we write this cephalic phase some in CRR. I hope we know CRR. What is CRR? Cephalic phase, in which your brain thinks a lot of food is gonna come and we're gonna have a nice meal. 20 to 30% of HCl is released in it. Okay, and it works via acetylcholine. Okay, and how it works is, it works that we have two type of fibers in parasympathetic system. We have pre-sympathetic, we have pre-parasympathetic and post-parasympathetic, and then we have the ganglion in the middle, and then here we have the spinal cord, right? They work via nicotinic receptors. Here they work via M1 to M5. We call that muscarinic receptors. Okay, what happened here is that this acetylcholine, it's released in this post parasympathetic fibers, and it goes and triggers this cephalic phase. And once cephalic phase is triggered, we release around 20 to 30% of HCl, and usually this M3, we use something called as M3 receptors on acetylcholine, right? So acetylcholine goes to M3 receptors, which are present on parietal cells, and then once they are positively stimulated, parietal cells upregulate their H plus K channels, proton pumps. They are called as proton pumps, and then we have enough acid. So that was a little bit about cephalic phase. Okay, so I hope we understand this part, right? Little bit part. Now the factors that can affect gastrin are increase and decrease. Let's talk about increase first. So enteral distension, we know just how it works. Protein rich diet, of course. Small amino acid can directly stimulate G cell via chemoreceptors, we know this. Gastrin releasing peptide, right? GRP, we'll talk about it in a second, right? Other thing is Ca, calcium, that is the exocytosis, right? Calcium leads to calcium is usually a signaling molecule. So if we have more calcium, we have more exocytosis of things. Okay, and calcium leads to increase in gastrin, because it leads to usually exocytosis of chemicals. Epinephrine. Now this epinephrine, right? Now this epinephrine is a part of sympathetic channel of your autonomic nervous system. Okay, autonomic nervous system. What is autonomic nervous system? Autonomic nervous system is a system. It has three branches. It has sympathetic system, it has parasympathetic system, and it has an enteric sympathetic system, enteric nervous system. That's it. These three can talk with each other, and they have different neurochemical, they have chemicals to talk with each other. So once we are on the sympathetic drive, once we are stressed, the adrenal medulla, what is the cortex? Cortex is the middle part, medulla is the outer part right here. For example, this is my medulla. I'm so sorry. This is my adrenal gland. It looks like a triangle. Right here on the periphery, we have medulla. On the cortex, we have different, different synthesis of chemicals. So medulla makes epinephrine. That's important. Okay, so epinephrine sympathetic adrenal medulla makes this, right? And then it's made right there, and then it travels in the blood, right? It travels and it reaches to our GIT, and then it reaches to our beta 2 adrenergic receptors. We have alpha 1, alpha 2, beta 1, and beta 2 receptor. We have four type of receptor. I don't want to explain each one of them, but try to understand we have four type of receptors, beta 1, and then this sympathetic goes and it binds with beta 2 receptors, and this beta 2 adrenergic receptor leads to increased level of camp and increased level of calcium. Now increased level of camp will lead to increased level of adenyl cyclase, right? We call it AC, and then AC will lead to more further pathways, and then eventually what will happen is that we will have a positive excitatory response. Okay, so that is how epinephrine can actually release gastrin, and then gastrin can release HCl. Okay, so this is why you should not take stress a lot of times. You should try to minimize your stress in your daily life. What about decrease? Well, excess acid, thank God, God has given us this negative feedback loop. If a lot of acid is right there in the duodenum, right, it will go inside this negative feedback loop and it will inhibit the release of further acid. This is excess acid. Somatostatin via D cells, right? D cells, they make somatostatin. This is amazing. We need this because it can stop almost everything. When we are going under this excessive hyper acidic condition in our stomach, actually D cells get activated via this ACH, right? Acetylcholine. What is acetylcholine? Acetylcholine is just a neurotransmitter that positively excites cell, right? So this is somatostatin, and it inhibits, and once it inhibits everything, the production of the acid goes down, and then we can feel alive again, right? Because we are gonna die because of this a lot of acid, right? GIP, that is inhibition peptides, right? I told you GIP was gastric inhibitory peptide, so it's inhibitory in nature, right? They are produced by K cells. I hate the name K cells because it just doesn't make sense. Okay, secretin release. Secretin release. So we know secretin is a natural antacid, right? And it's released by the brush border of S cells in the duodenum. They are produced when they sense some acidic chyme is arriving from stomach inside the duodenum, and duodenal brush border, they sense that, they sense the presence of acid. They tell the pancreas to start making a lot of bicarb, and then we have neutrality in the duodenum, and then we are happy. So I hope we understand all of them. It's very easy. It's nothing to really stress about. So this was our gastrin family, right? What about the actions of gastrin? Well, gastrin usually works by CCK-B receptors. This is very important, okay? Now, how it works? Well, CCK-B receptor, usually we have two type of action from CCK-B. CCK-B can lead to increase gastrin and increase HCl and decrease HCl. Okay, depending upon the position of this receptor, the movement, the result can be changed, right? Now the action of gastrin works via CCK-B receptors, right? And here what happens, right? I will talk about it. LES, this is lower esophageal sphincter. It is present right here. This is your esophagus, this is your food pipe, this is your stomach, this is your duodenum starting, right? This is your small intestine, right? And this is your big intestine. Right here is your lower esophageal sphincter. This is where your food is supposed to pass through, right? If this sphincter is really weak, what will happen is that the content of the stomach, the acidic chyme can go back, and it can lead to squamous cell carcinoma, right, of the esophagus, right? Okay, so via CCK-B receptors, right? So we have 1, 2, 3, 4, 5 functions of this gastrin hormone. Number one function is that it increases acid, very obvious, via CCK-B. And because CCK-B receptors are present on these parietal cells. Okay, I am not saying that all of the CCKB receptors are present on parietal cells. The CCKB receptors that are present on the parietal cells, they can increase HCl. That's what I'm saying. Other thing is it lowers means reduces. Not here is not there. Lower esophageal sphincter contraction. It helps in contraction of the lower esophageal sphincter. I told you that the hormones that are secreted in your stomach will always push the food in your duodenum, and the hormone that the hormone that are produced in the duodenum or in the intestine, they want to stop the food. They are against the motility of the food that is coming from stomach into the duodenum. So this is very important. So it makes sure that the LES is really well contracted, right? And it prevents the reflux, right? And that is protective in nature. Okay, reflux of what? Reflux of the food that you have eaten, and you have acid, right? But amazing thing is that if you have any crisis of acid in your stomach, you will vomit, and this contraction will go loose, and then you can vomit a lot of acid outside your body. This is one of the triad of alkalosis, right? It's triad. It means we have increased as alkalosis of blood, we have H ion secreted in urine, and we have vomit. So this is right. And even a lot of CO2, we lose a lot of CO2 from our lungs. That also causes a lot of alkalinity of the blood. Okay. Prevents the reflux. Okay. Increase the gastric motility, because it wants food. It wants, this hormone gastrin wants food content to move from stomach to the duodenum, right? Increase level of pepsin. Pepsin means it's a hormone that digests protein. So increased level of pepsin, of course. What about trophic action? Now this is a lot of people, they forget about the trophic action. It stimulates GI tract mucosal growth. It is a protective factor for the acid. It protects your mucosa from the acid. How? What happened here is that this gastrin, it works via CCKB mainly, and when the CCKB, right, it works via CCKB, and it makes sure that ECL is producing histamine plus some growth factors, right? And this CCKB also acts on parietal cells and stem cells. So this complex called histamine and growth factor is released from ECL, right? And the parietal cells and the stem cells are also regulated well. So this growth zone of GIT, right, the stem cell, right, that is the growth zone, is positively stimulated via gastrin. So gastrin can actually help us to heal our stomach after an episodic ulcer. Okay, so this is very important. I hope we understand. Let's talk a little bit about LES, that is lower esophageal sphincter. So the things that can increase the contraction or increase the tonicity. Tone mean how tight your LES. By the way, LES, it's a physiological sphincter. It's not an anatomical. There is no flap there. It's just how your esophagus is made. For example, I will make a diagram. This is your starting of the buccal cavity. This is your pharynx or larynx, whatever you want to say. This is your, imagine this, okay? And now I'm starting this esophageal, right? And right here is my stomach, okay? Right? This is my stomach. Okay, stomach. This part is my sphincter, right? Here anatomically there is nothing. There is no flap here. But how it really works is that this is a pressure system like that. Your esophagus works like that. There is a pressure difference between this part and this part. So we have this physiological sphincter right here. Okay, so that was it. Amazing though. So now we understand this. So LES. So the things that can increase the contractility and the tone of LES is acetylcholine via M3 receptors, right? M3 is muscarinic 3. We have M1, 2, M5 that are present on the parasympathetic fibers, parasympathetic post-ganglionic fibers. Okay, so ACH works like this. We have gastrin. Gastrin is this hormone that is produced by G cells, and it also tightens your. We have mortallin against for the movement. Substance P also works for contracting this, right? LES. Then we have proteins, and then we have alpha adrenergic agonist. Okay, I will talk about it. So the LES is positively affected by these things, right? We'll study each and every hormone in detail soon. LES is negatively affected. It relaxes. It means it opens up via VIP, NO, that is nitric oxide. Secretin, that is the diurnal hormone, right? I told you it wants to stop. CCK, surprisingly, usually CCK is released in the diurnum, right, from the eye cells, right? So CCK. Now you would think that gastrin and CCK, they come from same family, but ironically CCK, it wants to relax the LES. It wants to send back the food into your buccal cavity, into your mouth. Progesterone, surprise, surprise. That's why a lot of women during their pregnancy experience their vomiting, emetism, whatever you want to call it, vomiting a lot of times, right? Alcohol, it weakens this tone, caffeine and smoke as well, and beta 2 receptors, right? So I hope we understand LES right now. Cool? Okay, so here we have cholecystokinin, pancreozymin. One thing I know that it releases these, it results in contraction of gallbladder. That's very important. So gastrin was working via CCKB, and cholecystokinin, pancreozymin, works by CCKA receptors, right? What happens is that when we eat something, food that is rich in fat or protein, this fat and protein is sensed by these eye cells that are present in the diurnum, right? And once this senses that you have eaten something fatty, it will lead to releasing this cholecystokinin, pancreozymin. Once this hormone is released, it will lead to this bicarb rich, pancreatic enzymes being released inside the lumen of the diurnum. So CCK, right, action is work via CCKA. It's a cholagogue, right? Cholagogue means it increases the contractility of gallbladder. Okay, let's go back a little bit. So cholagogue, it increases the contraction of gallbladder, and cholerectic is just, it increases bile synthesis. So chemicals that are called cholerectic are these bile synthesis. Bile salts, secretin, and vasostimulation. We have three type of pathways. Bile salt, they work via direct stimulation of hepatocytes. Secretin works via indirect, and vagal via neurotic. Not neurotic, neural, right? How secretin works? Secretin goes on these cholangiocytes, right? These are bile duct epithelial cells, and via CFTR, they are upregulation of the CFTR channel and leads to rich bicarb bile flow. Vagal stimulation leads to cephalic stage, more ACH on gallbladder, smooth cell contraction happens. Smooth cell can show contractility because they have these things. Okay, I forgot what the name of this thing. So smooth cell, what makes smooth cell special is that smooth cell are built for contraction actually. They don't have striation like skeletal muscles or cardiac muscles. They have these actin and myosin abundance, right? And they have these calcium control system, right? So Doug, if you have smooth cells, you will see contraction, right? So okay, let's go back to our cholecystokinin, pancreozymin. Okay, it's a cholagogue. It means it increases the contraction of gallbladder. It releases bile, right? It relaxation of sphincter of Oddi. Okay, important. GI action. GI action means gastric action, of course. Augments the action of secretin. It means it increases the action of secretin. How? Because what happens here is that this CCKPZ, it acts via acinar cells. Acinar cells are these exocrine glands that produce a lot of saliva and a lot of exocrine hormones, right? We'll talk about what they make. So CCKPZ acts on these acinar. It makes them more sensitive to secretin by increasing the CAMP levels and increasing the calcium level signaling, right? When we eat something, when food is eaten, it goes, right? We have more acid and we have fat present. Now this leads to eye cells being released, and then CCPZ and gallbladder releases this HCO3 bicarb rich enzyme, right? And then this increased acid can release to secretin, and then I hope the story makes sense, right? So secretin and this CCK, that is CCK, I hope it makes sense, right? Okay, stimulates the intestinal and colonic motility. Okay, cool. Inhibit, stops gastric emptying because it's an intestinal hormone. I hope we have built this concept by now. Intestinal hormone want to stop gastric emptying inside the duodenum because they are not ready. They will want to have more time. The CNS action of CCK, that is cholecystokinin, is that it reduces the food intake by increasing the satiety via vagal afferents. So vagal afferent means it goes to the cell body. So vagal afferent goes to hypothalamus. There are CCK receptors in the hypothalamus, and then we feel that we don't want to eat, right? There's two type of system: ventral, and then they're like oraxin nucleus, and then we have ventral nucleus. Ventral nucleus is for when we feel satiety, when we are full. Ventral nucleus wanna not eat, makes us not eat, and oraxin nucleus wants to make us eat. So just remember that. Implicated in anxiety, that's very important. How anxiety and CCK are, so what happens? CCK goes and binds with CCKB receptors, and these CCKB receptors are present on the limbic system, and it works via dopamine and glutamate pathway. So it can change the dopamine and glutamate. Both dopamine and glutamate can change your moods easily, because glutamate is a it's an excitatory neuro chemical, and dopamine has D1, D2, D3, D4, D5, and they work by a camp inhibitory, camp stimulatory, and camp Q. This not camp, sorry, G inhibitory, G stimulatory, and G this. So okay, in short, they can mess with your dopamine and glutamate. Another thing, analgesia. Analgesia means ability to handle pain. So CCK can work via endogenous in this endogenous opioid receptor things, right? And this can increase or decrease the opioid effect of your endogenous opioids. You make your own opioids, right? So it works also for CCKB. So usually I just remember that CCKB receptor are present in the brain usually, because on hypothalamus I've seen it, on this limbic system I've seen it. So CCB, I think about brain. A is for different things, So PZ. What is PZ? Pancreazymin. It stimulates the secretion of enzyme. Enzymes are these these fat lipase, right? These proteinases, right? Enzyme lipase, electrolyte rich pancreatic juice. Electrolyte rich means HCO3 rich. Okay, so that was CCKPZ and Gastrin, and we would think that they work, they are from same family, but they are a little bit different. So it's very important that we understand their function and their relation with each other. Okay.